I will give you a lecture about mycorrhizal definitions. So going back to the roots in mycorrhiza. I'm sharing you my slides. I'm a research professor in the University of Tartu and jointly also in the King Saud University of Saudi Arabia. I will speak about mycorrhiza definition, mycorrhiza types, use of mycorrhizal traits for metastudies, and also fungal root database. So first, about mycorrhiza definition. I have been giving lectures in mycorrhiza for almost 20 years. And I could say that mycorrhiza is a symbiotic organ between a root and the fungus. The origin of the term mycorrhiza dates back to 1885, when Professor Albert Bernhard Frank described first mycorrhizal structures and started to use this term. Mycorrhiza is defined as a differentiated plant fungal root association in which both partners are potentially in mutually beneficial relationships in terms of nutrition. It is important that in mycorrhiza, plant fungal complex structures evolve, and these act as interface for transportation of organic carbon from plant to fungus and transfer of water and dissolved nutrients in the opposite direction. Mycorrhiza also involves other mycorrhizosphere bacteria and protists. There are also other benefits to plants besides access to nitrogen, phosphorus, and micronutrients. These include better access to water in small soil pores, defense against pathogens and herbivores, and chemicals and heavy metals. Additional benefits to fungi include better access to deep groundwater, relatively stable growing environment, and storage of excess nutrients in case of avascular mycorrhiza. Going to more scientific side, the definition remains the same, but since we know so many exceptions and extra cases in avascular mycorrhiza, we should say that mycorrhiza is a differentiated plant-root fungal interaction in which both partners are potentially in mutually beneficial relationships in terms of nutrition, but because of multiple morphological and functional exceptions, 3 out of the 4 following criteria need to be fulfilled. So these criteria are the structure, potential nutritional mutualism, phylogeny in both fungal and plant side. It is especially critical for understanding avascular mycorrhiza, and therefore the structural criteria includes avascules, which include both arum type and Paris type structures. When considering potentially nutritional mutualism, here we include also mycoheterotrophs, in which this nutrient and carbon flow has been just reversed. In fungal phylogeny, we consider potential AM symbionts to be Glomeromycota and Endogonomycetes from Mucoromycota. For plants, AM plants include vascular plants that have not lost essential functional genes, but these plants include bryophytes and other non-vascular plants. So, for example, the loss of genes has occurred in many instances in vascular mycorrhiza, and one of the largest cases loss in the order Brassicales, in which many of the families, including Brassicaceae family, and which also includes Arabidopsis and Arabis genera, has lost all genes necessary for vascular mycorrhizal formation, and thus I think we can consider this group as secondarily non-mycorrhizal quite safely. These recent definitions have been published and more explained in two relatively recent publications together with Professor Mark Brandrett. This slide illustrates the main four types of mycorrhiza. First of all, arbuscular mycorrhiza. Here you can see the essential arbuscules, but also there may be vesicles, and there may be intra and extracellular hyphae, which are not always very important for the arbuscular mycorrhizal structure itself, but at least arbuscules as the interface are always very important. Vesicles and intercellular hyphae can occur also without arbuscules, but in these cases we would rather consider these as glomeromycotan endophytes. The second main type of mycorrhiza is ectomycorrhiza, in which Hartig net and fungal mantle are the key structural elements, and in particular through Hartig net, carbon and nutrient transfer occurs. The third type is ericoid mycorrhiza, in which generally endophytic fungi colonize root of ericoid plant cells and form dense hyphal coils in the epidermal root cells. We can see this fungi as domesticated endophytes because taxonomically they overlap with true endophytes of other plants. Fourth main type is orchid mycorrhiza. Here one could argue that quite often orchid mycorrhiza are not beneficial for fungi, but they can be beneficial only for the plant. In orchid mycorrhiza, again, intracellular structures called pelotons evolve, and older pelotons are digested within the plant root cells. You can see also there is quite a long list of subtypes of ectomycorrhiza, and there is also the fifth type of mycorrhiza called dysanotoid mycorrhiza, but to be honest, nobody really well understands what it is about because there has been so little research so far. Structures comparable to true mycorrhizas are also liverwort mycorrhiza-like structures. Well, liverworts don't have roots, so whether they have mycorrhizas or not is a matter of definition. And when talking about mycorrhizas, we also have to consider root endophytes, that grow inside roots of all plants. So especially for arbuscular mycorrhiza, there are several alternative definitions and classifications. So first, arbuscular mycorrhiza was called as vesicular arbuscular mycorrhiza, which was then in 1990 turned into arbuscular mycorrhiza because it was found that vesicles are not that important structure in arbuscular mycorrhiza. Quite recently, just 5 years ago, also so-called fine root endophytes in the newly described genus Planticonsortium were considered as arbuscular mycorrhizal. These narrow hyphal fungi belong to Endogonomycetes in Mucoromycota, and in very many occasions also true arbuscules are observed with these fungi. Again, whether or not to include hepatics as mycorrhizal is a matter of taste, but there is a good alternative term called mycorrhizal. Very recently, my colleagues in Tartu, namely Buena et al., proposed that arbuscular mycorrhizas would entirely lack arbuscules. Well, in that case, a nutrient exchange between plants and fungi would be strongly inhibited, although we can argue that non-nutritional benefits may still remain. This definition would also include all plants that have roots, because especially hyphae of Glomeromycota grow very well intercellularly in other plants, such as non-mycorrhizal mutants of real arbuscular mycorrhizal plants, naturally non-mycorrhizal plants, ectomycorrhizal plants, orchids, but also Glomeromycota can grow throughout the thalli and rhizoids of mosses. So this would make this lack of arbuscular mycorrhiza more look like endophyte. Endophytes are defined as symptomless and mostly commensal plant root microbe interactions in case of roots. Next, we also have to understand that when we are compiling databases about mycorrhizal traits, there are very many errors in these data sets. We have estimated that in well-studied Nordic tree species, around 10% of all data points are incorrect, and the abundance of these incorrect data points ranges from 0 to 48% in some cases. On average, across various databases, it has been estimated that 10 to 33% of all data points may be incorrect. Especially this number refers to Mycoflora database, in which the major inconsistencies have been found between arbuscular mycorrhizal and non-mycorrhizal states. The errors are also non-random. They are more frequent in cases where plants have a bit more strange root structures, or if they are very strongly colonized by endophytes. So these all can be misidentified as arbuscular mycorrhiza. And sadly, errors are also accumulating from one database to another. So for example, one of the very first mycorrhizal traits databases was generated by Trappy. It was especially for ectomycorrhizal host plants. Although the authors acknowledged that not all their data may be right, because these were based on fruiting body observations and links to trees, then many other authors picked up these reports and lost this error message accompanying this paper. So from Trappy, for example, these reports were taken up by Harley and Harley, although they also included warning against using these data as a golden standard. These were also picked up by the Mycoflora database, and also in the very commonly cited FungalTraits database. Also, it's quite unfortunate that those potential errors are very difficult to distinguish from real biological phenomena. For example, also very recently, it was reported that a small herb, Pulsatilla patens, in Poland may form both ectomycorrhiza and arbuscular mycorrhiza. This was found in many places across Poland. The authors provided compelling evidence in terms of cross sections, and they also provided molecular identification for both the plant and fungal partner. So essentially, it would be perfect if such reports of very suspicious cases are backed up by such illustrations and molecular confirmation. So what happens if mycorrhizal type or status is misassigned? It may be very vital for ordinary people. So imagine a poor African family who goes mushrooming in a wrong forest, and they are not able to find their favorite amanita mushrooms for dinner. So it means that this family is probably starving. But when coming to science, then misassigning mycorrhizal types in plants may result in misinterpretation of carbon cycle, nitrogen cycle, many different ecosystem services. Also, there may be very strong miscalculations of evolutionary processes, because in evolutionary analysis, every single misassignment may generate a new case of evolutionary event, and all these states and ancestral states may be hence misinterpreted. Also, there may be major misinterpretations in plant community dynamics and also population dynamics. Examples and reasoning for these are provided in two publications, Brandrett and Tederso, 2019, and my paper with other colleagues in 2019. And what happens when we relax the mycorrhiza definition by allowing arbuscular mycorrhiza plants to be without arbuscules? It may also result in misinterpretation of soil carbon and nitrogen cycle. Also other ecosystem services, evolutionary processes, and also plant community and functioning. It is essentially important when there are studies about carbon and nutrient flow between plants or between fungi and plants in play. And here on the right are examples of studies for which there are assumptions that from plant to fungi and the opposite way of nutrient flow is occurring. If these assumptions are not correct, then these studies would be actually wrong. As there are so many erroneous reports, there is certainly a question how to use these generally crappy data. First option is to ignore. It would mean no dataset curation or no expert opinion. It may result in very strong misunderstanding of the ecological data, because simply many things wouldn't work or are biased. And since the errors are non-random, then also modeling this error may be quite difficult and may not do this job properly. The third option is to curate the databases to weed out errors. It means adding expert opinions and modeling the errors inside the databases. Also, these are vulnerable to non-randomness in the data. How to deal with missing traits data? The problem is a big one, because when we want to compare mycorrhizal traits globally, the data are very unevenly distributed. And this is also one reason why we cannot ignore this data sparsity. We just can't exclude all species that lack empirical observations. One option is to extrapolate phylogenetic, especially genus level data to genera. Genera tend to be quite strongly conserved in mycorrhizal traits, especially mycorrhizal type. These extrapolations are not that secure at the family level, because different genera may have quite different patterns. We also have to consider that commonly there is some intrageneric variation, and variation at the family level can be much stronger. Besides this, we can also extrapolate to patterns in extreme communities or life forms, such as aquatic plants or carnivorous plants, but this is conditionally applicable only for non-mycorrhizal plants. So to overcome these issues, we generated the fungal root database led by Nadezhda Sochilovskaya. Fungal root database is an empirical database of plant mycorrhizal associations. Now it includes more than 36,000 records from nearly 15,000 plant species. It also includes abundant soil and geography related metadata, such as soil pH and geographical coordinates. It also highlights potentially unreliable studies, which are considered as studies with more than 20% of conflicting records to other empirically recorded data. We also provide and allow providing expert opinions for genus level recommendations and about the validity of the data. So in our case, we use 2/3 of consensus when recommending one plant genus, for example, to be either arbuscular mycorrhizal, non-mycorrhizal, or of any other type. In the new second version, we have also more possibilities for third-party opinions and especially confirmation of arbuscules as structures in the roots. Our fungal root database was very recently criticized by my colleagues in Tartu, Bueno et al. They stated that arbuscules should not be critical for arbuscular mycorrhiza, and it is mainly hyphal colonization that matters. As we showed before, essentially in this scenario, all root-bearing plants should be considered as arbuscular mycorrhiza, and many ecosystem processes would not work as these have been described. Bueno et al. Provide example that Arabis hirsute should be considered as facultatively arbuscular mycorrhizal because a few studies have found a few arbuscules. But the fact is that Arabis species, as other Brassicaceae, lack all genes necessary for arbuscular mycorrhiza formation. So any formation of true arbuscules initiated by the plant is very unlikely in all species of Brassicaceae. Bueno et al. Also claimed that fungal root mistranslates original reports. Actually, fungal root provides all original reports and also remarks on the methods of determination used in each case. Bueno et al. Also state that fungal root has taken most of their data from previous compilations. Again, this is not true. The data are directly obtained from original sources along with the metadata. Bueno et al. Say that there are strong issues with highlighting unreliable data, but we have quite clearly described that we have used 20% of conflict threshold relative to other empirical reports, and this is quite common practice in other studies to have some kind of thresholds to consider data that needs to be taken critically. Bueno et al. Also say that our expert opinions and criteria for genus level extrapolation are untransparent. Well, the truth is that all expert opinions are justified in specific data fields in the database, and that these rely on 2/3 of majority rule for the level of general. And what is surprising that Bueno et al. Actually used their own expert opinions in their studies, but these and their all underlying data are actually unavailable and thus unreproducible. So by all means, the paper of Bueno et al. Leaves an impression that not all authors understand what has been written, as they have twisted several facts indicative of either carelessness or malicious intent. It is also very cynical that they challenge credibility of fungal roots, while their own mycorrhizal traits database and expert opinions therein are completely untransparent. So with this, I conclude, and I want to thank my colleagues in University of Tartu mycology team, the fungal root core team, especially Nadja Sochilovskaya and Mark Brandrett. So thank you for your attention, and I'm happy to take your questions.